Hello everyone, habari zenyu saseni? Welcome back to this lecture on pulmonary surfactants. For today I would like to start a slightly different. Let's listen to the story of Sarah and Noah. Let's start with the story of Sarah and Noah. Sarah has just given birth to her baby boy, Noah, a few weeks earlier than expected. She's relieved to see his little face, but her joy is mixed with worry. Noah seems to be struggling with each breath. His tiny chest moves rapidly up and down, his nostrils flare with every inhale, and there is a faint bluish tint around his lips and fingertips. The nurse calls this respiratory distress. Sarah's heart sinks as the doctor gently explains. Noah's lungs are not fully developed yet because he was born early. His lungs lack something called pulmonary surfactant. Confused and anxious, Sarah wonders aloud. But what does surfactant do? And why will it cause him to struggle like this? That's where we are going to start our lecture, in today's lecture. We're going to explore what pulmonary surfactant is, how it is formed, and why it is essential for every breath a newborn takes. Hopefully, at the end of this, we can be able to answer Sarah's questions. So what is this pulmonary surfactant? So it is a lipid protein complex that lines the alveoli of the lungs. We do know the alveoli is the site of exchange of gases between the environment and our body. We know we exchange oxygen for carbon dioxide at the alveoli. We do know surfactant is very efficient for reducing surface tension, and it prevents the lung to collapse during expiration. We also know it has other functions such as immune defense. It plays a big role in immunity, where it has proteins. The proteins it contains, they help recognize and bind pathogens, and so it plays a very big role in innate immunity. So what is the lipid composition of surfactant? So as we said, it has both lipids and proteins, and the lipids make up 80 to 90% of the surfactant. Of all the lipids present, 70% of them are called phosphatidylcholine, the one we call dipalmitoyl phosphatidylcholine. So many books will abbreviate it as DPPC. This is the main component, the main lipid in surfactant. However, there are other lipids which appear in low concentration, like phosphatidylglycerol, phosphatidylinositol, phosphatidylserine, phosphatidylethanolamine, and sphingomyelin. We said it also contains proteins, and the proteins found in surfactant have four major ones: SP, which stands for surfactant-specific proteins, A, B, C, and D. SPA-A and SPA-D are hydrophilic, meaning they are water-loving, and they play an important role in immune defense. However, SPB and SPC are hydrophobic, and we're going to see SPB is very important in the formation of lamellar bodies, which we find in the alveolar type 2 cells, which make surfactant. SPC plays a very important role in improving surfactant deposition and its function. And also we find that SPB, in addition to its function of formation of lamellar bodies, also has antimicrobial properties. So these proteins are the lipids, are the ones that make up what we know as pulmonary surfactant. So how is surfactant synthesized? So we do know the surfactant are synthesized in the alveoli by a type of cell called alveolar type 2 cells. So in the alveoli, if you remember from your anatomy, there are two types of alveolar cells, alveolar type 1 and alveolar type 2. But for this lecture, we are going to focus on the alveolar type 2 cells that synthesize surfactant. So the first step is the synthesis of the molecule of the phospholipids, and this synthesis occurs in the endoplasmic reticulum of the alveolar type 2 cells. So the phospholipids are transported through the Golgi apparatus to lamellar bodies, okay? And then these lamellar bodies are like small vesicular structures that store surfactant, and they are present in the alveolar type 2 cells. In the lamellar bodies, we have a formation of the surfactant lipoprotein complex, okay? And surfactant is released from these lamellar bodies through the process of exocytosis from the apical surface of the type 2 alveolar cells. So we can see the important organelles in surfactant synthesis are the endoplasmic reticulum, the Golgi apparatus, and the lamellar bodies. Let's look a bit more in the details of the biosynthetic process of surfactant. So the first step is the formation of glycerol, which forms the backbone of the glycerophospholipid dipalmitoyl phosphatidyl choline. So where does glycerol 3-phosphate come from? Glycerol 3-phosphate comes from glycolysis. If you remember, we talked about glycolysis. This is where we get glycerol 3-phosphate, and it provides the backbone for surfactant. So we have the enzyme glycerol 3-phosphate acyltransferase, which adds a fatty acid to the first carbon of the glycerol molecule to form lysophosphatidic acid. Usually this fatty acid is palmitoyl-CoA. Okay, and so when it is added to the first carbon of glycerol 3-phosphate, we end up with lysophosphatidic acid. In the next step, we have another enzyme called 1-acylglycerol 3-phosphate acyltransferase, which adds a second fatty acid to the second carbon of glycerol. Remember, the first carbon had OH, and now we've added a fatty acid there. The second carbon also had a OH, and we're adding a second carbon, a second fatty acid there. And now the third carbon, remember, you're attached to phosphate. That's why it's called glycerol 3-phosphate. So this enzyme, AG part, adds the second fatty acid, usually again palmitoyl-CoA, to form what we refer to as phosphatidic acid. And the phosphatidic acid is the parental backbone for many phospholipids. The phosphatidic acid can be dephosphorylated by phosphatidic acid phosphatase to form diacylglycerol, and this serves as the precursor of triacylglycerol. But in our case, diacylglycerol will be used in the next steps for the synthesis of DPPC. Let's see how this happens. So now, the next thing that needs to form is the choline. So choline is first phosphorylated. Remember, choline can come from diet or from recycled surfactant. So this, from whichever source choline comes from, at first it is phosphorylated to give us choline phosphate. And then the enzyme choline phosphate cytidyl transferase adds this choline phosphate to cytidine triphosphate to form our CDP choline. Okay, so this is a two-step reaction. First, phosphorylation of choline, and then conjugating it to CDP to form CDP choline. So the formation of DPPC will start with the combination or condensation of CDP choline with diacylglycerol, and this is catalyzed by CDP choline diacylglycerol choline phosphotransferase. Okay, and they come together, they create what we call phosphatidyl choline. The phosphatidyl choline can be added to another palmitate molecule. We have an acyl group added, and I said commonly it's palmitate, to this to give us our dipalmitoyl phosphatidyl choline, or our DPPC, which we refer to as surfactant. And we say this one forms as much as 70% of the lipids that we find in the molecule surfactant, with the compound surfactant. So once surfactant is formed, we've said it has released into in the lumen of the alveoli by the process of exocytosis. We actually refer to the apical lumen. So what happens to surfactant? Does it live here forever? No. After some time, some of the surfactant is taken back into the type 2 pneumocytes in the process of recycling. And the recycled phospholipids can be reassembled again to form new surfactant. So this process is very important because it prevents overaccumulation of surfactant. It allows the lungs to maintain an adequate level of surfactant, and also it reduces the cost of synthesis without constant renewal or constant synthesis of new molecules of choline, diacylglycerol, and those other things. However, some of the surfactant is degraded, okay? And this one is done by the very effective alveolar macrophages. So what they do, they can take up phagocytose, the surfactant, and break down it into its respective components. And these components can then either remove from the lungs, or they can be used by the alveoli cell, alveolar macrophages for its metabolic needs. I'm imagining it will use the glycerol for energy production, okay? So these are the two things that happen to the surfactant. It can be recycled or degraded. So what are the primary functions of pulmonary surfactant? We said there are two main functions: to lower the surface tension, and the proteins, they play the host defense role against pathogens. Okay, so usually when we think about the lung tissue, the surfactant helps us to reduce the surface tension at the air-liquid interface in the alveoli, and so this prevents collapse at the end of the expiration. So this is very important. That's why the children, the newborn children, but even us adults, require surfactant in our lungs to prevent this surface tension. So also we know the proteins, as we said, they can help in host defense by binding to bacteria and viral surfaces, and this binding helps the macrophages and other cells of the immune cells to clear the pathogens. It also opsonizes the bacteria and makes them tasty. Remember, the process of opsonization was a process which proteins were coated around the foreign organism, and this made them more appealing to the macrophages and monocytes, which took them for phagocytosis. Another effect of the proteins have been found is that they can also have direct antimicrobial effects, where they increase the membrane permeability of their bacteria and fungi. And we know increase in membrane permeability will cause leakage of the cytoplasm, or in entering into the cytoplasm of these organisms different compounds, which will cause death or damage of the bacteria. So this causes the bacteria of the fungi to be destroyed. So these are some of the ways that the host defense proteins, or the proteins SPA, B, SPA, and D, help to protect the lung against pathogens. Let's look a bit more at this concept of surface tension. So we do know that in the alveoli, we have a space where the air enters. When we breathe in, the air enters into this space. And within this space also, we have some water molecules, because like gases like oxygen will dissolve in water, which will facilitate their diffusion from the gaseous aspect into the bloodstream. Okay, so once we breathe out, so when we breathe in, it's like a balloon. When we breathe in, it's like a balloon, and the alveoli opens up. But the issue comes out when we expirate. There's a possibility that the water molecules, which we said they have very strong cohesive properties, to be attracted to each other. And you know, water molecule is very cohesive and they pull each other close together. And so when they pull each other, it will be like a collapsed balloon. And they're likely to bring the surfaces of the alveoli together. However, this is not done like this, because the alveoli lines the inner layer of the surfactant lines the inner layer of the alveoli. So what does that mean? It reduces the cohesiveness of water molecules. And so even when we breathe out, the two sides of the alveoli do not come close together. Okay, they do not come close together like the balloon without air. especially if a balloon, you think about it, a balloon without air and then it has water. It comes close together and sometimes it's even very hard to blow. So this is what this diagram is showing us. If there was no surfactant here, it will require a lot of pressure to blow, to breathe in oxygen, to breathe in air into this alveoli after expiration, because it will have collapsed. But luckily for us, in the presence of surfactant, even when we breathe out air, these two ends do not come together and the pressure required to push air in is not as much. And I want you to relate with this thing we saw at the beginning of Noah. He was struggling to breathe and he seemed like his chest kept on raising because he required a lot of pressure to push air into his alveoli because he did not have surfactant. So the surface tension is the elastic force created by a fluid surface that minimizes the surface area via cohesion of liquid molecules. So I think all this we have explained. I hope you have understood how water increases the surface tension here. And when we expirate, the alveolar surfaces can come very close together if there was no surfactant. Understanding how surfactant still works. So we say that surfactant, where we find it is, you can see this is the alveoli. Inside here, the blue part is where we have air, and we can see the cells lining the alveoli. If we just take a bit of magnification, we can see the air layer, and then we have the water layer. And so this air diffuses across the pneumocytes into the capillaries. And we can see here the big ones, type 2 pneumocyte cells. However, what happens? In between the air and the water, we have the molecule of surfactant. And remember, if you think about the molecule dipalmitol phosphatidylcholine, it has an amphibolic nature. Amphipathic nature, not amphibolic, amphipathic. Amphipathic means it both has a hydrophobic end and a hydrophilic end. So the hydrophobic end is the one that comes direct into contact with the air, okay? While the hydrophilic ends, they come into contact with the water. So it separates direct contact between the air and the water. So what does this mean? Remember, these water molecules, they love each other. They have very strong cohesion, and they're the ones that increase the surface tension. So surfactant being here, it is able to reduce the cohesion, the hydrogen bonds between the water molecules, between these water molecules and the water molecules on this other side of the alveoli, such that when you breathe out, these water molecules are not very strongly attracted to this other end because there's a layer of surfactant. So we say this action breaks some of the hydrogen bonds in the water, causing it to lose its spherical cell shape and lowering the surface tension within the alveoli. And so this is very good, especially when we breathe out, meaning this space will not completely close like the balloon without air. We will have some space. And so when you're breathing in, bringing in more air, we don't have to force a lot of air through that balloon that has completely collapsed because it will be partially open. So this is how surfactant helps to reduce the surface tension and facilitates the process of gas exchange. So what happens if we have reduced production of surfactant or we have dysfunction of surfactant? So the common one is what we saw in Noah, where we had the respiratory distress syndrome, where we have insufficient production of surfactant, okay? So the surface tension cannot be reduced and we are going to see the child presents with respiratory distress. We also have other instances where the mutations in some of the SPC proteins, which are autosomal dominant mutations, or we can have autosomal recessive mutations of SPB or ABA-CA. This is one of the proteins that facilitates the movement of the phospholipids into the lamellar bodies. So if there is a mutations of this, they will cause problems with production of surfactant, and so we'll have difficulty in breathing. And usually the cases can be quite severe and they might require lung transplantation, especially in the neonates. And then in adults, we can have an acute respiratory distress syndrome, where we can have altered surfactant composition and function. Probably if an individual has been exposed to gases like sulfur dioxide, which can damage the surfactant, and so over a period of time, the individual fails to breathe because the surfactant has been interfered with and they have acute respiratory distress syndrome. We can also have a problem, as we saw, it was very important to either recycle or degrade surfactant because, again, an overabundance of surfactant can cause another problem. One of the problems is what we know as pulmonary alveolar proteinosis. Here we have increased accumulation of alveoli because of the deficient of a receptor that is found in the alveolar macrophage that facilitate its taking up or phagocytosis of the surfactant to degrade it. So if this accumulates, we'll have impaired gas exchange and can cause again respiratory distress. Interestingly, elevated levels of SPA and SPAD can be used as markers to assess the level of lung injury and pulmonary fibrosis. We know when there's continuous damage to the lung tissue, it fibroses and it loses elasticity, and the fibrosed tissues like a scar tissue, it is not able to do oxygen transfer, it cannot transfer tissues. So the level of SPA and SPD can be used as potential indicators of disease severity, especially in H1N1 infection, viral infections. Expecting that when we have infection, probably we'll have increased production of SPA and SPD, so they have immune functions. We have some respiratory infections that can also impact surfactant. Such of them are like bacterial infections called by Pseudomonas aeruginosa. They can inhibit surfactant production, and they can increase the rate of degradation of SPA and SPAD, which we know have immune functions. And then some viral infections like the respiratory syncytial virus and adenovirus can also decrease the expression of the surfactant proteins, and this can disrupt lipid trafficking. So I hope it is clear whether we have excess of abundance of surfactant or underabundance of surfactant, we are likely to get a problem. So let's come back to the case of Noah. What is this neonatal respiratory distress syndrome, and why does it happen? I'm sure we have a lot of questions. So as we had seen, it is a distress syndrome that is commonly occurs in newborn, and it presents within hours after birth or immediately after delivery as they try to make the baby breathe after delivery. And it is more common in preterm infants, so the children who have been born before 39 weeks. However, it is a bit rare in term infants, although it can also occur. And the more smaller the premature, the more severe the disease. Of course, in addition to the features of difficulty in breathing, we had described in Noah, but we can see also brain function can be interfered with when we have low oxygen, okay? And remember, at this time of birth, we really want to make sure the brain of the child is not damaged because this can bring problems later on in life. So to understand formation of surfactant in the neonate, let's look slightly in a bit of embryology. We're not going to talk about embryology. So the fetal lung maturation progresses with increasing gestational age. So we have 5 stages of lung development, but the stage I want us to focus about is the alveolar stage. This ranges from 32 weeks through childhood. And it is in this alveolar stage where the type 2 pneumocytes that make alveolar mature and they start producing surfactant. So that's why earlier in the slide we mentioned the younger the child is, the more severe the respiratory distress is, because we can see in these particular stages, we have not even started making the alveoli. We just have canaculation and growth of the bronchi and division. Okay, so at alveolar stages where we have maturation of the alveolar and type 2 pneumocytes mature and they start surfactant production, and this is at around 32 weeks. So this could also advise why would the child would have to stay in utero longer, and also inform us of the risk of a child born before 32 weeks. So these type 2 pneumocytes, they form around 7% of the internal surface of the alveoli. Okay, so there are quite a few, but just not as much. And they are big cells whose cytoplasm contains the lamellar bodies we had mentioned earlier. Okay, and we say these are organelles that store and they secrete surfactants. And they are such to appear around 22 to 24 weeks of gestation, and they increase at around 32 weeks of gestation. And when you start seeing the type 2 pneumocytes in the fetal lungs, they indicate that these lungs are starting to mature. So there's an old biochemical test that they used to use to assess if a child is mature, although it's no longer used as much today. However, this was measuring the ratio of lecithin to sphingomyelin. It's called lecithin sphingomyelin, or the LS ratio. So as we have seen, before the 28th weeks, the alveolar type 2 cells are not made, are not prepared, are not formed well. And so the alveoli are the lung tissues making a lipid called sphingomyelin. And this sphingomyelin does not play a big role in gaseous exchange. Remember, even at this time, the lungs of the baby are filled with fluid because the gas exchange is being done at the placenta. But as the child continues maturing and the alveolar type 2 cells are forming, we start seeing that the type 2 starts synthesizing lecithin. And as the child matures, as the child grows older, the amount of sphingomyelin reduces and the amount of lecithin increases as we form more lamellae. The interesting thing is that both sphingomyelin and lecithin are present in amniotic fluid. And so through amniocentesis, a sample of the amniotic fluid can be taken, and through chromatography, we can measure the amount of sphingomyelin versus the amount of lecithin. So the LS ratio is used to evaluate whether the fetal lungs are mature. And so as you expect, the younger the child, we see the sphingomyelin will be higher. And as the child matures, the sphingomyelin amounts will reduce and the lecithin will increase. So we really want that ratio to be over 1.8, where we have more lecithin and the lung, it indicates maturity of the fetal lung. So these days, this test is not commonly done because of the risk associated with amniocentesis. However, it could be indicated because of poor dating of the pregnancy. We don't know how long, or when delivery is planned between 32 to 39 weeks, and we don't know whether the lungs are mature. So how is the management? How will this NOAA be managed? Just a slight bit of hope. So usually management of neonatal respiratory distress syndrome should start even before the mother gives birth, using antenatal corticosteroids. So these antenatal corticosteroids have been found to decrease the incidence and the severity. So we know we have some mothers who can have indications to give birth, or for the medical practitioners to remove the child before term. So when we know this is planned, we can start giving the mother antenatal corticosteroids. So these corticosteroids have been found to cross the placenta, and they can stimulate that process of lung structure development, especially the alveoli. They also stimulate the maturation of the type 2 pneumocytes, which synthesize surfactant. They also help to modify the composition of surfactant such that we can have higher levels of phospholipids, especially dipalmitoyl phosphatidylcholine. And finally, the steroids have been found to make the surfactant more effective at reducing surface tension and contribute to better respiratory outcomes in the premature infants. However, as now that Noah has already been born, he can be managed through respiratory support, some surfactant therapy, and just overall care of the premature infant as we await the alveolar type 2 cells to mature, and hopefully his lungs can mature and gain back their regular function. As we end this lecture, we say a prayer for Sarah and Noah, and we hope for the quick recovery of Noah. Thank you.